Hi, my name is Angela Bawardi, and I'm going to be talking about the hepatitis A virus. So, its official name is hepatitis A virus or hepatovirus A due to its hepatoviral classification. It has four common names, and that's hepatitis A, Hep A, infectious hepatitis, and traveler's disease. So, its infectious agent is hepatitis A virus, and its common name is hepatitis A. Throughout this presentation, I'm probably just going to be saying Hep A. So, to the left, we have its full taxonomy. Classification, so the domain is Rubivaria, kingdom is Orthonavirinae, phylum is Picornaviricoda, class is Picornaviricetes, order is Picornavirilis, family is Picornaviridae, genus is Hepatovirus, and species is Hepatovirus A. There's also Hep B, C, D, and E that are similar to Hep A in different ways. So Hep B and C are in the same domain as Hep A, and Hep C is also in the same kingdom as Hep A. Hep D is unique because it develops and is dependent on Hep B virus. And Hep E and A are the same, such that they're both transmitted through contaminated food and water. So Hep A was first identified in 1973 as a member of the picornavirus clade. The members of the picornavirus consist of any family of Picornaviridae of single-stranded RNA and causative agents of polio, encephalomyocarditis, hepatitis A, foot and mouth disease, and hand, foot, and mouth disease. So Hep A has an icosahedral geometry as shown to the image on the right, and it's a non-enveloped virus with a diameter of 27 to 32 nanometers. It's the smallest and simplest RNA virus, so it makes it more difficult to identify its icosahedral geometry in comparison to other members of the picornavirus. And it has two main components, and that's the viral proteins, which we'll look into further later on in this presentation, and its positive sense single-stranded RNA. So there are four strains of hepatitis A, which are B, C, D, and E. So Hep A, C, D, and E will have RNA genome, but Hep B will have a DNA genome. Hep A is the most mild and least hostile infection. This doesn't mean that it cannot cause life-threatening issues, but fatality is very rare. So it's an acute liver disease. Its average incubation is around 2 weeks to 28 days, and it's transmitted through contaminated food and water. So it's more prevalent in underdeveloped countries like parts of Africa and Asia. Hep B is an acute liver disease that can become chronic, and its average incubation is about 70 to 80 days. It's spread through semen, blood, bodily fluids, and dirty needles. It can also be spread from an infected mother to her baby during childbirth. Hep C is the most common and probably the most well-known strain. There's no treatment for Hep C right now, but 95% of cases can be treated with antiviral therapy. It can cause chronic infection. Average incubation is about 50 days, and it's transmitted through dirty needles. So, like, for example, blood transfusions or sharing needles with another individual if you're doing drugs or something, and improperly sterilized medical equipment. Hep D is unique because it is dependent on a chronic Hep B infection, and that's why it's known as a superinfection. Its average incubation is about 30 days, and it's an acute infection that can become chronic. Hepatitis E is similar to Hep A because it's the most mild and least hostile strain. It's also transmitted through contaminated food and water. It can cause acute liver failure in pregnant women, fetal loss, or even death. So for Hep A, the liver will inflame suddenly. So after ingestion of contaminated food or drinks, the virus will enter the bloodstream through the epithelium of the oropharynx, which is the throat or the intestine, and the virus is carried to the liver by the blood and reproduces within liver macrophages. These are hepatocytes or Kupffer cells. They're also known as liver cells. The virions are secreted into the bile and then released in the feces. And hepatitis A virus is excreted 11 days in large amounts before symptoms are actually seen or before IgM antibodies are seen. So each capsid has around 60 duplicates of viral protein 1, 2, and 3. So viral protein 1 and 3 are visible on the surface of the capsid, and it basically gives its conformational immunodominant antigenic site. And RNA inside the hepatocytes, the RNA genome is released from its protein coat, and it's translated by the host cell on protein-producing ribosomes. So even though Hep A is part of the picornavirus clade, it requires an intact eukaryotic initiating factor 4G, and that's what this is over here, for initiation and translation. So the necessity for this initiating factor leads to a longer adaptation period to grow hepatitis A virus cell cultures because the virus must incompetently compete for cellular translation. So the WHO organization estimates around 1.5 million cases yearly, and in high endemic areas like parts of Africa and Asia, this is due to contaminated foods and waters or inadequate sanitation. In more developed countries like the U.S., this is due to imported foods like pomegranates from Turkey. And then there's an increase in hepatitis A infections in travelers and homosexual intercourse between men. So there was a decrease in hepatitis A infections. However, starting from 2012, there is now an increase. So in 2012 and 2013, there was an increase due to imported pomegranates from Turkey. In 2015 and 16, it increased again by another 44.4% due to imported foods. 2017 and 18, there was an increase because of an increase in drug use and opioid crisis, as well as more individuals that were homeless. So in 2017, there was 91 deaths. In 2019, there was an increase because of fresh blackberries in 11 Midwest states, Wisconsin or Illinois. And in 2021, so like this year, of November 12th, there's around 42,920 cases. Of those cases, 61 people were hospitalized and 402 people have died. So it's seen that the Hep A virus is now exceeding death rates of HIV, malaria, and tuberculosis, with around 1.4 to 1.5 million cases yearly. So there are different endemicities in different regions of the world. So in less developed countries, Hep A infections peak in a younger age. In developing countries, it peaks in teenage years. In developed countries, it peaks in younger adults. So the risk of being affected with hepatitis A is not age-related or gender-related, but it's more of if you travel a lot, or if like you're a laboratory scientist that handles the virus itself, if you're involved in risky intersexual courses, and if you basically do not have good hygiene. So the first appearance of HIV was in the 17th century, and it was associated with military campaigns. The first outbreak was in Virginia in 1812, and the virus was most prevalent amongst the Union troops, with around 40,000 cases reported. Hep A affected individual troops during World War I and II because of inadequate sanitation and the virus transmitted by fecal-oral mode of transmission in the field. At this point, there was around 16 million cases, and there was an outbreak of hepatitis that was correlated with the yellow fever vaccine, and it was stabilized with human serums. So at this point, there was around 50,000 developing cases, which sparked researchers to want to do more research on Hep A. So the research showed that one form of hepatitis spikes much quicker than the other, and this was the serum hepatitis. It had a incubation period of around 90 days, where epidemic or infectious hepatitis had a shorter incubation period, which is 18 to 25 days. More research was conducted, and it showed that although some soldiers had immunity for hepatitis, they were still able to develop a second infectious hepatitis, so this gave rise to the terms hepatitis A and B. So hepatitis is seen in the feces of individuals with a virus. So after you ingest something that was contaminated, the virus will enter the throat or the intestine. It can be transmitted from person-to-person contact with someone infected, or if an infected person prepares foods or drinks for an uninfected person, because no matter how small the residue of fecal matter is, you will still be infected by hepatitis. Symptoms do not begin immediately. There's around a 2-week incubation before symptoms actually begin. It's usually the virus usually infects an individual with the fecal-oral route. There's another mode of entry, which is contaminated water. So if, like sewage water, or if the water is not treated adequately. To prevent hepatitis, there are many different ways. So like vaccine, there are 2 doses that are taken 6 months to a year apart. So after the first dose, you'll receive immunity 2 weeks into it, and it will last for just a little while. But after you take the second dose, it'll last for 20 years to life. It's very safe and highly effective. There's also natural immunity, so IgM and IgG antibodies can be present in the bloodstream. So IgM is the first antibody that's seen if someone has Hep A. IgG is seen in the bloodstream after Hep A infection has subsided. Now this provides a secondary immune response. There's also healthy habits, so like personal hygiene, proper sanitation, avoid sharing personal belongings. You must keep in mind that alcohol-based sanitizers are not enough alone to not be infected with Hep A, but it can decrease chances of being infected with Hep A. And also, if you were exposed to Hep A, you can still receive the vaccine 2 weeks after exposure to the virus. So Hep A is caused by viruses that affect the liver cells, which we said were hepatocytes or Kupffer cells. It causes inflammation and damage to the liver, so that the virus attacks the Kupffer cells, and this inflammation is part of the response, so that the immune system activates to fight the virus. The inflammation is usually temporary, but it can last months or years, which becomes chronic, and it will cause a decreased processing of vital nutrients and eliminating toxins in the body, which is basically the liver. And if no treatment is conducted, it can lead to cirrhosis of the liver, which is scarring. So over here we have like a healthy liver, fibrosis, cirrhosis, which is the scarring over here, and then this is cancer, which we really don't want. So Hep A is dependent greatly on the humoral immune response. So there are IgM and IgG antibodies. IgM is the first antibody that's seen if someone has Hep A, and IgG antibodies are seen after the person has Hep A, basically. Hepatitis antigens are only in the cytoplasm of hepatocytes and Kupffer cells, and their antigens are linked with 27 nanometer virus-like particles found in the liver and feces of animals. So the antigens can be said to be antigenically interrelated. So Hep A is mostly diagnosed by symptoms and clinical tests, but blood tests are most common because individuals can be asymptomatic. So blood is drawn from the arm and it's sent to the lab, where the blood test detects antibodies like IgM and IgG. If IgM is present, that means the person is currently infected with Hep A. If IgG is present, that means they have a natural immunity or artificial immunity by vaccines to Hep A. If the blood test shows a positive result for hepatitis, imaging tests of the livers can be conducted, or a physical exam to look at the color of the sclera or tender abdomen, because Hep A can cause jaundice because it reduces the adequacy of liver function. If there is still not enough information, a CT scan or MRI can be done to suggest for liver cancer. And if this is still not enough information, a liver biopsy can be conducted to look at the level of cirrhosis or scarring in the liver. So right now there is still no treatment for Hep A, but there are many different ways to prevent from obtaining Hep A, which is the humoral immune response, where the liver will usually heal in 6 months with little to no damage, natural active immunity, or artificial active immunity like vaccines. So right now, the health department is taking a closer look at New Jersey because as of November of 2021, there was a potential hepatitis A outbreak. A Starbucks employee was working in Starbucks serving customers drinks while she was in her infectious period. So right now, the Starbucks right there is closed until all employees are fully vaccinated for Hep A. WHO organization is also trying to eradicate hepatitis viral-like infections, but more specifically B and C by 2030, because Hep B and C death rates are starting to exceed the death rates of HIV, malaria, and TB. There's also Andrea Cox, where she explained that HIV, malaria, and TB deaths have started to decrease since 2008, whereas Hep B and C death rates are starting to increase. And then Jia Cao is trying to increase implementation of hepatitis B vaccines, especially for pregnant mothers in their second and third trimesters. And finally, WHO is trying to raise awareness of hepatitis virus, increasing healthcare sanitation, increasing availability of vaccines, and making vaccines cheaper for everyone to get. So thank you for listening. These are just my references, but I'll see you guys in class.